This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 729, recorded on March 9th, 2021. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent and everybody else. I spent the better part of this afternoon sitting on my porch. I have we live on the 8th floor of a condominium, and the weather outside is absolutely spectacular. It's the first nice day of the year, I think.
We still have snow in our backyard, you know. It's still piled up because there's so much. Matt Snowplow is still out there, but the weather has taken a turn for the better. Also joining us from Madison, New Jersey, Brienne Barker.
Hi, it's great to be here. I second everything that Dixon just said. It is 63 and sunny and gorgeous out.
From Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 57 Fahrenheit, 14 Celsius, partly cloudy. Really nice day.
Your name lends itself to that radio Spindler, you know, the sports announce type thing. It's really good for that. Okay. And from Austin, Texas, Rich Condit.
Howdy, 73, cloudy. I mean, good temperature, but I'm, sort of into sunshine.
We have a guest today.
You're from the Sunshine State.
Our guest today is from the University of Maryland in Baltimore. He is back for the 10th time on TWiV. Matt Frieman, welcome back.
Thank you very much. Happy to be here.
10
Good
TWiVs.
to see you again, Matt.
It's a lot.
10 TWiVs. 10 TWiVs.
The most recent number here is 287, and that's 2014. So did you have the 7-year itch? Come back.
That's right. It's called a pandemic.
Turns out coronaviruses are back in the news a little bit. So
like every decade, right? We have a coronavirus outbreak of some kind.
No, thanks.
No, thanks?
No, this is enough.
Well, we agree with that. This one is enough. Anyway, Matt, welcome back. The first episode was 65, and that was called Matt's Bats. That was back when you were sampling bats. Do you remember those days?
Yeah, that was when what year was that? Was that when I was here, right? I think. It wasn't when I was a postdoc. It was when I had started Maryland.
I think you had just started in Maryland, yeah.
Yeah, absolutely. No, that seems like 4 lifetimes ago now.
It does. And then episode 75, 171, 175, 204, 207, 239, 258, and 287. Wow.
Lord. You guys got all too big for me. You didn't need me anymore. It's okay.
I was thinking of that. We basically ignored coronaviruses for a long time. I think that was it. And now we're being punished.
I want to ask why has Matt not aged a bit and we all have? What's up with that?
I certainly feel much older than I probably should.
He's still a young guy.
You don't look a day over the time we first met.
Good thing.
So Matt,
yeah, it is.
Back in the beginning or the end of 2019, beginning of 2020, did you see this coming, or were you suspicious? Were you surprised? Give us your thoughts on the very beginning.
So the beginning for me, I put this slide in my talks lately. I put a tweet out on December 31st at like 8:30 in the morning, and it was the ProMed post of this unusual pneumonia cluster in Hubei province, and I think I wrote that I have to pull up. I think I wrote like probably not SARS, but worth looking at or something. And that was December 31st, 2019. And basically all day on, day before New Year's, I spent translating with Google Translate Chinese news reports, looking for anything that was on this. And by the end of like 10:30 or 11 o'clock at night, there was a report saying that there was this large number of people in this hospital in Hubei. And that's the next tweet. And, ever since then, it's been kind of full force. And I don't think anyone really thought we would be doing what we're doing now at the time. If they say they thought this was going to turn into this global pandemic, I think they certainly are lying. But it's spun up very fast, as you know, right? You've ran through all the histories in your episodes, but I think by the end of January, everyone really knew that this was something that was not going to go away very quickly.
Yeah. I mean, we should say, even though you've been on 9 episodes before, you are a coronavirus expert. You've done your whole career on coronaviruses, right?
Right, absolutely. So my postdoc, my PhD is actually in yeast genetics from Hopkins. So I got a good background there on just basic molecular biology techniques. And then I went to Ralph Barrick's lab for my postdoc at UNC, where I started working on SARS. And then I started here at Maryland in 2009, starting with SARS, and then MERS in 2012. And then, I think one of the really kind of amazing things for us and for the field has been the ability for all of us to who have coronavirus experience previously to kind of help guide a lot of the therapeutics and the companies and really trying to understand what this virus is doing based on how we can compare it to the previous viruses. And I think that has really helped both inform the therapeutic side, but also kind of understanding about what's going on.
That's been a really collaborative effort. Is that right? Amongst the different coronavirus labs?
Certainly. I mean, we talked at all kinds of NIH and WHO meetings. There's been a really large consortium of people, obviously around the world, working on these viruses, and we really do share a lot of data as it's coming. I have a lot of colleagues from both Ralph's lab when I was there that are now other places, and we all talk all the time to kind of share things as we're finding them and ask questions and share reagents and really get ahead of the game as much as we can.
So when you
so often when
hold it, Dixon.
Sorry, go ahead.
so
Yeah,
you mentioned the ProMed post and kind of having an idea that things were interesting by the end of January. When or like what type of data said to you, oh, this is different. This is going to be something.
I really think it was so my lab reminds me this all the time. The first week of January, we literally had a lab meeting and we're like, we're not going to work on this virus. Everyone's so busy. Like it's going to be a little thing. And it's not, they always tease me. And obviously we were, a couple of weeks later when the cases kept going and there was more news coming out of China that this was spreading. And certainly by the end of January, when the first cases showed up in the US, it was like, okay, this is obviously what we're doing. Also at that time, we were getting lots of calls from companies about doing vaccine work and antibody work. And I think we're going to talk about some of that today, but it was certainly something we were going to be doing. And whether it was going to be 500 cases or, whatever, the numbers it is now, we were still going to be working on this virus. So it became very obvious pretty quick as the cases kind of were accruing that this wasn't going to be something that was going to stay in China, like people had thought initially.
Okay, Dixon. Now you're muted.
Yes, all I wanted to say was that, no, I'm not muted. I'm unmuted. All I wanted to say, Matt, was that very often when something like this happens, you get people jumping on the bandwagon, and you've got so many people on it that you're not sure of who the bandwagon was. But today we're talking to the bandwagon. You are it. So that's a real privilege because you've seen this thing from the absolute beginning, and there's a lot of wisdom associated with being.
Well, there's certainly a lot of groups working on this virus now, and they have in the last year. There's really an amazing amount of experience outside of coronaviruses that have been incredibly useful, whether it's on giant drug screens, whether it is on antibody design or antigen design or vaccine design. All of these other things come into play. And, it's been, while at moments I may be annoyed at the people that are getting attention and getting papers published here and there while we're trying to do really conscientious and clean experiments and people are doing one-off experiments and getting into science. It's okay. It's okay. We're not a flash in the pan kind of coronavirus lab. So we're going to be here for a while. And luckily we've been able to find our way through all of this and really help out where we can. I mean, that's what our goal has been in the lab, is to get as many projects into the clinic as possible. So partnering with companies that have actionable projects and really can get into trials and get things out. And that's really been a lot of the focus and time spent this year has been working on those kind of projects.
Matt, I do want to talk about Novavax and what you've done with them. But before we do that, let's get something out of the way. This is something we've dealt with, as you know, for the past year here on TWiV and many other people. this idea that this virus came from a lab, simply won't die. And just last week, a bunch of scientists signed a letter. And unfortunately, one of the scientists is Jean-Michel Claverie of Giant Virus fame, calling for an all-out investigation into its origins. How can we put this to rest, Matt?
I don't think it's something that can be put to rest. It's not really something that we can say is not lab escaped or not engineered or anything. There's certainly evidence of both sides of at least, or, suspicions of something going on in a lab that may have led to this. It doesn't really, to me, it doesn't really matter because the virus is out there and that's what we're dealing with now. There doesn't seem to be any hard evidence at all that this is something to do with any kind of engineered or escaped thing. But again, it's kind of answering an unanswerable question at the moment. And it doesn't really concern what we do, change what we do on a daily basis of how this works. So I try to stay away from the origin questions and deal with what we have now.
Yeah.
Can I ask you a bat question?
Sure.
I know that's sort of origin-y, but so I know that on one of your past TWiV episodes, you were talking about bats and looking at bats. So what types of coronaviruses have you detected in bats, and have you detected them in places outside of China or Southeast Asia? Do you know anything about the coronaviruses in the bats in the US, for example?
Yeah, so I think one of the initiating events for my first TWiV episode was this project we had with when I was a postdoc with Eric Donaldson, who was a grad student and then a postdoc in Ralph's lab. He's now at the FDA, and he is. We had a project working with a bat ecologist in Western Maryland who was capturing bats, and we were basically sequencing viruses that came out of bat fecal samples. And we identified 2 coronaviruses out of that, which we called Appalachian Ridge coronavirus, or 1 and 2. And those were alpha coronaviruses. They were different than the beta coronavirus lineages that we've seen for SARS-2 and SARS-1 and MERS, but they certainly are out there. And, even in the US, there are bat coronaviruses that are circulating around bat populations, I'm sure. There has not really been a huge network of detection and screening in bats in the United States for that kind of project, but it's certainly something we're interested in getting into again in the future. But whether there are bat coronaviruses like we're seeing SARS-like bat coronaviruses that you're seeing in China, I doubt that they're in the United States. But viruses are viruses. Stranger things have happened, so they could be out there, I guess.
Now, Novavax, you've been working with them clearly over the past year. How did that start? Did you know them before the outbreak?
I did. And so this is one of these great, to me, this is a nice thing about science, is that after MERS emerged in 2012, we started working with Novavax on a MERS vaccine. And so we have a couple papers with them using the same technology, almost exactly the same system they're doing now for SARS-2. They used a vaccine, they produced a vaccine against the MERS spike. There's been certainly some knowledge differences on this, how to generate a better antigen for spike since then, but essentially the same exact way they did it in 2012 is what they're doing now. And it's basically, they use insect cells or moth cells. I don't think anyone who works on SF9 cells knows they're from moths, other than since Novavax started being in the news, but they use baculovirus infection of insect cells expressing a full-length spike protein. At that time it was for MERS, and then extracting it from the membrane of the insect cells. And the interesting thing about it is that since it's a full-length spike, it has the transmembrane domain as part of it. And so you get this, when you extract it in the detergent, you get a little floret of spikes. You get 4 or 5 spikes that come together because they're sticky C-terminus, all 6 together as this hydrophobic spot. They call it a nanoparticle. I call it a floret, but they're things, so we'll call it a nanoparticle. It's not like a nanoparticle in the sense there's a bead in the middle with spikes off of it. That's not what it is. It looks like a little kind of a jack, like you're playing jacks as a kid. That's what it looks like, with 5 or 6 little spikes stuck together in the middle. And then they mix it with their own adjuvant, which they own an adjuvant called Matrix M1. And we did the same thing for MERS. We vaccinated mice with that mixture of Matrix M1 and MERS spike nanoparticles. Produces a great antibody response in mice. And then you can, at that time, we're using the adenovirus DPP4 challenge model in mice to make them permissive to MERS. And they're protected from infection. And so that vaccine got also used in a cow project. So there's a company called SAB, which they have transchromosomic bovines, TCBs, where they have an extra chromosome. Have you ever heard this story? They have an extra chromosome in them, so when they make antibodies, they make human antibodies as well as cow antibodies. Huh. It's amazing. It's great.
It's the Velocicow.
It's similar idea, exactly, like the Regeneron idea, right? Except it's cows, and the antibodies don't go at high levels in the milk, which would be beautiful if it did, but they go into the blood, and so you basically put a cow on a giant plasmapheresis system, and you cycle off liters of serum per month, and it makes and so they use that vaccine that we had been made it for MERS. I had somehow connected to this company called SAB, and told them about the vaccine project, and so they use that for MERS. And so basically we worked with them on this MERS project for several years, published a couple really nice papers. It sat on the shelf. They actually did a phase 1 trial on the antibodies. And in January of 2019, when this was all coming up again, the Gail Smith, one of the head scientists at Novavax, emailed me and basically responded to an email I sent him like 4 years before, saying, "Hey, we're going to make a coronavirus vaccine. Do you want to be part of this with us again?" And I said, "Absolutely." And then it's been basically going since then. So I really like that we could cycle back to these collaborators, whether it's Novavax, whether it's Regeneron, who we did monoclonals with for MERS before. That's really, I like that aspect of science, is we can come back to these stories and knowing that there's good partners and good expertise in these companies, we can be the basic science angle to this as well.
That person. So Matt, eventually
I don't know about that. As most of the last year, I just felt like a guy with a BL3 and a pipette. So that's pretty much what we did.
I was wondering what your role was in a lot of these projects, whether you're the virus neutralization guy or what.
So I hope that we had some scientific input in these projects. I think especially for Novavax, they started with several different constructs of spikes. And so we were able to both prove that their spikes were functional by doing neutral inhibition assays right away for their protein production line, as well as do some very quick and early neutralization assays in different constructs from mice to know which were going to work the best. And then I think you had Barney Graham and Jason McClellan on already.
We've had Jason on, not Barney.
Jason, yeah. So Jason, obviously his pre-fusion spike form discoveries over the last several years have been incredibly important for all of these vaccine designs. And so his figuring out the mutations that you could use for the pre-fusion spike is in the Novavax vaccine as well. And it's really, I think it's in every single one of the warp speed, all the major vaccines all have the pre-fusion spike, I believe. And Novavax also has mutations in the furin domain, the furin site, which allows for a more stable particle. So that was one of the things we tested early on, is to see which ones were better. And they have all this beautiful chemistry on their particles to show that they call them 2P, 3Q mutants with the pre-fusion and the furin cleavage site mutants were a better, more anagenically stable particle. And that goes into the stability of the vaccine, where it doesn't need -80 or whatever cold chain like the Moderna and Pfizer vaccines do. You can really use it as refrigerated normal coolers and it's fine. So, I mean, all of these little bits added up over time. And it's really, I think this is just the good science. This is how these little parts work together, and it's all additive at some level.
So Jason actually told us that AstraZeneca did not use a pre-fusion spike in their vectored vaccine. And
okay.
I wanted to ask you what you thought about that. I mean, they've had some issues with their vaccine. Do you think possibly it has something to do with not using the pre-fusion stabilized?
I don't think that's as important in the virus vectored vaccines, because it's more important when you're making a lot of the protein like Novavax was doing. Okay. When you're expressing it inside a cell, whether it's the mRNA or the chimp adeno version, I don't think the pre-fusion stayed as mutations are as important. It certainly was important to make the particle after formulation better. And one of the, I think it's in the first paper we have Novavax last year for their phase 1 data, they show a bunch of chemical dispersion properties of their particle. And it really makes this beautifully tight, small, stable particle that is resistant to a lot of temperature and salt shifts and all kinds of other chemical parameters that make it a better antigen.
And the MERS vaccine that you talked about, it was not pre-fusion stabilized, right?
Right. At the time, I don't believe that we knew anything about the pre-fusion mutations. I don't remember when Jason's first paper came out on the coronavirus spikes, but that was just the normal one. MERS spike also does have a furin cleavage site in it, so that was already in there as well. So as if it would have been a ramped up procedure where they had to really produce in large quantities like they have to now, maybe that would have been something that we would have got to, but it wasn't on our radar at that point.
So if is there any need for a MERS vaccine, say for camels in the Arabian Peninsula? Would this one be a good candidate?
We have certainly talked about getting a MERS vaccine into camels in Saudi Arabia for years. We've worked on several different vaccine projects. Another one with Matthias Schnell at Thomas Jefferson and Reed Johnson at the IRF on a rabies, killed rabies vectored MERS vaccine, where you get rabies glycoprotein as well as spike from MERS on the surface of the particle. So you get a dual vaccine. Right. I think it's great. It works great in animals. But all of these would be very useful in camels, I think, in Saudi Arabia. But the numbers of cases of MERS have obviously not ramped up over the years like we see for anything else. So it's not as much of a urgent problem there at the moment.
Yeah, I'm really curious about the production. I mean, moth cells, they must be growing hundreds of thousands of liters, right? Do they have big vats that they do this in?
Yeah, absolutely. And I think they have, I don't remember how many different production companies they're working with now to make the doses, but they're on, there's one in India, one in China, one in the United States, and I think one in Europe as well. And so they have they have 1,000-liter bioreactors. It's these incredibly giant production lines. And that, as much as we work in the lab, I'm a bench scientist. There's no way I could possibly, I don't understand at all what it takes to produce something at scale like that.
It does rise.
Getting it fill and finished, getting it through the FDA, like all of these things. There are really important parts of this that are well outside of just knowing that the vaccine works in an animal or even a human arm for small production scale.
So
so the reason why they use it, I'm sorry.
It seems to me that the baculovirus technology is, common to a number of different products. So it seems to me that this is a technology that you could tool up another company to adopt if necessary, as opposed to the more novel, I'm making this up, I'm looking for comment, more novel like mRNA technologies, where I would think that tooling that up from scratch would be a bigger problem. Is that a fair statement?
I think so. I mean, I have to say, and I think I've said this to a lot of people last year, I certainly did not think that the mRNA vaccines were going to be as good as they are. I don't think anyone other than people at Moderna and BioNTech would have believed that they worked as well as they did. I was certainly poo-pooing them at the beginning of this. And part of it was because of the massive production that you need to get this many doses. And it just wasn't possible. And it was really over the last year where they've figured out ways of doing huge scale of these mRNA vaccine productions. And so it will change the rest, I think, the future vaccine designs for both emerging and other vaccines that people are developing. It's certainly going to be a game changer in that sense. At the beginning of this, when there was Operation Warp Speed was developing, and Moderna and Pfizer and AstraZeneca were getting this money, we're sitting in the lab. I have no stake in Novavax, so I don't. I just wanted things we did in the lab to be in people's arms as fast as possible. we were waiting and waiting and waiting for them to get a vaccine company that had kind of a standard technology. And so having Novavax as a protein subunit technology certainly was one of the more standard ones that they funded, luckily. And since then, Novavax has gotten CEPI money and Gates Foundation, DoD, and a bunch of other funding mechanisms. So it certainly helped drive the company, both stock, which I own none of, and their ability to really just develop doses and get production lines up and running in various countries around the world. And so I do think that it's a really good vaccine. It works great. And there's certainly interesting things that came out of the UK trial and the South Africa trial. And now they're in phase 3 in the United States. So I think it's the next one kind of online to come out of their clinical trial and hopefully get an EUA approval.
So do you see any differences in types of immune responses compared to some of those other vaccines, like, different types of T cell subsets or different types of antibody responses or things like that?
Yeah, that's a great question. And I think that the one of the things that has been, both a blessing and a curse, I guess, through all of this is that there are different labs running assays for all of these companies. And they really are apples and oranges. I mean, we all are doing, you guys have hit on the neutralization before. We do a neutralization assay in my lab, which is a standard microneutralization CPE-based assay. Other people are doing pseudotype assays. And there's been a lot of concordant studies, or at least attempts at concordant studies over the year, at least on neutralization. And it's still kind of all up in the air. They're close to being the same, but it really depends on how you do these assays to get numbers. And that's just, that part doesn't really bother me. I think that as long as it's consistent and the same groups are doing the things for consistently in the assays, then it's fine. I don't have much of a problem with it. I think the B and T cell side is really interesting. And I think one thing we don't know is which kind of parameters for especially T cell responses are going to lead toward long-term memory response. And I'm not an immunologist. I won't pretend to be. You can have Shane Crody on, and he can give you T cell based information for 3 hours. And I certainly think that neutralizing antibody responses are at least 90% there of what we look for. And I think time will tell if whether you're presenting your antigen internally of a cell because of mRNA or a virus, or externally as a subunit vaccine, which one of those works the best long term. I think that's up in the air, and I certainly don't know which of those is the right answer.
My sense from looking at the papers and the Novavax literature is that their adjuvant is an important feature of this whole thing. The Matrix M adjuvant, which is a proprietary, as they describe it, thing. But it's, as I was reading up on it, it itself is saponin presented in the form of a couple of different cholesterol-based nanoparticles. And I'm wondering if you have any sense of, first of all, do they have a real clinical experience with this adjuvant outside of these trials, and whether this adjuvant really has any particular advantages over other adjuvants that impacts on the efficacy of this vaccine?
Absolutely. So the Matrix M adjuvant is their own. It's a saponin-based adjuvant. It's from this, I don't remember if it was the New Yorker or the Atlantic. There's a really funny article interviewing the people, the heads of Novavax, where they say at the beginning of this, they called their supplier, which is in Chile. So it's from this Chilean soap bark tree where they get the adjuvant from. And they called them early on and said, listen, we need as much as you can make. And so they've been producing this obviously continuously. But it certainly is, it's an adjuvant they've used for all of their vaccines. Whether we did it, the same vaccine for MERS, they have a whole series of them over time, RSV as well. And it really leads to this infiltration of antigen presenting cells, and then activation of B cells and T cells and APCs that really help drive the neutralizing antibody response and hopefully T cell response over time. So we've done studies where you don't have the adjuvant, and you do have the adjuvant, and it really does enhance the protection both in animals and also the antibody response.
Does that create more reactogenicity in the individuals getting the vaccine?
Yeah, no, not at all, and which is interesting. So I think that the data from the trials has shown that I think the Moderna and the Pfizer vaccinations have a bit more reactogenicity than Novavax has seen in their trials so far. Obviously, the U.S. phase 3 trial hasn't read out yet. The data on the UK phase 3 trial isn't published, but at least from their reported reactogenicity, it hasn't been a problem. I don't know if how many of you are vaccinated, but I got the Moderna vaccine. The second dose of the Moderna vaccine knocked me out for about 24 hours. So as more people in the U.S. get it vaccinated, I think it's going to be interesting to see how widespread these responses are. But I certainly would be happier to be vaccinated for 24 hours. I mean, to knocked out for 24 hours after a vaccine than to get SARS coronavirus 2 infected.
That second dose knockout seems to be pretty common. Happened to my wife. I shrugged it off.
Yeah, tough guy.
Yeah.
So Matt, you said that phase 3 is ongoing in the U.S. Is that right?
Yes.
When is that expected to be over? Do you know?
I think it's sometime in April is kind of the expected readout based on the trajectory of the trial. They've already enrolled all 30,000 or so participants in the trial.
Right.
Yeah.
As of February.
Yeah. So it was a quick enrollment, which was amazing. I think it's 30 something sites in the United States is where they're doing the tests. The Center for Vaccine Development here at Maryland is running one of the test sites as well. And so there is, we're certainly excited to see the results in the US to see how it works. And both against the strains that are circulating now, including B117, which in the UK trial, it works fantastic against. And then we'll see if we run into the 1351 as it emerges in the United States too.
So, the press release that came out some time ago, this was all the UK and the South African results, right? So, if you look at that, what do you find good? What are the good parts that we should focus on?
So the good parts of it are that in the UK response, I'm just pulling up the numbers. In the UK response, the number is like 96.5% protect or vaccine efficacy against the original Washington 1 strain. When you add in B117, I think it goes to 89.2 or something percent protective. So those are obviously great numbers and way better. I mean, I never thought any of these vaccines were going to work at this level in humans at all. I don't think anyone could have expected this. I'm flabbergasted at how good these vaccines have been working. But so in the UK trial, I think they work like they should, like we expected to them, based on the really high neutralizing antibody levels that we saw both in mice and in macaques and in baboons previously during the last year. The South Africa trial is the one I think that is more interesting readout, where they ran into 1351 emerging, the B1, B.1.351 running, emerging in South Africa. And I think that was the first vaccine trial showing evidence that that South Africa variant was really going to be a problem for some of these vaccines.
So the South African one is what they call a phase 2B. What makes it 2B as opposed to 2 or 3? Do you know?
Oh, I know the numbers were smaller. So that there was only 4,400 people, right, in that trial. So that is certainly, I mean, I think that was a small aspects of it. I think that's one of the major differences. I'm sure there's clinical trial classifications between 2B and 3. But I know it was definitely smaller. The other thing that's interesting in that trial is they had HIV positive and HIV negative people in the trial, which had been an exclusionary criteria for several of the vaccines before then. And so it was really interesting to look at those differences just in the standard comorbidities of people out in the world, as well as running into this new South Africa variant as part of it. They also had 30% of the trial participants were previously COVID positive. So they were seropositive already. And so that was all of these little bits added to this kind of really interesting data set that came out of that really small trial, but I think really important trial.
Yeah.
So I have one other question about the U.S. trial, actually, the U.S. study. And you may not know the answer to this, but this question has come into me and I don't know the answer. So I'm figured. I'm just checking. A friend of mine is actually in the Novavax trial. And she was sort of annoyed because apparently she was required to get the doses in 2 different arms.
Oh.
And I wondered if you knew anything about that. She had some trouble with her pipette arm because she had to get her second dose in her pipette arm.
I don't know. I did not know that at all. I don't know why. I don't know why they would do it that way. Maybe. I have no clue.
Okay.
I haven't seen any reports of other trial data from Novavax saying that there was some weird reactogenicity if you got the dose in the same arm again or anything like that. I don't know that at all. Did she have any reactogenicity to the vaccine? Okay.
Yes.
Yeah.
So
Matt,
go ahead.
Go ahead.
Okay.
Well, mine's kind of a change of subject, or back to when you said when your lab decided that you were going to go this way. I was wondering how many of them were already BSL-3 trained, and has it changed the trajectory for anybody's thesis work or their postdoc projects in a major way? I mean, how does it work for a student's project, for example?
So we, so at the time, at the beginning of 2020, we had 4 people in the lab. And we now have 10, including me. So it certainly has changed the lab aspects a little bit. I had a grad student working on an influenza vaccine project and a vaccine adjuvant project. And Rob Haupt, who will be graduating this June, an exceptional grad student from the Army, actually. He's on loan to us from the Army for his PhD, and he's going back to them after he's done. So his project, we kept it as a flu project, but he certainly became he'd already been trained in the BL3, so he became one of our mouse kind of drivers of the mouse pathogenesis studies. He did all the Novavax mouse work, so he's a co-first author on the mouse data out of those studies. We gained a new technician. We gained 2 new grad students who started rotations and then stayed in the lab. So they obviously are doing COVID projects now. And they had the strangest rotations in the history of graduate school, I think, where they joined the lab in one in, I guess, March or so, and one maybe in May. And they kind of jumped on a moving locomotive as we were kind of going through. Both of them have been instrumental in driving what we're doing in the lab. So that has been kind of remarkable to watch, to see everybody come together. I rehired my first grad student in the lab, Carly Page at the time, now Carly Dillon. She came back as a project manager to help me wrangle all these bits and pieces of contracts and companies and academic partnerships together. And so it's really been a remarkable change in the lab. Also, you have to remember at the time, in around March 17th or something, Baltimore shut down for quarantine. And so we kept going and everything around us was closing, which was a certainly strange experience to be in the middle of. And people started leaving the building, and we basically had an empty building to ourselves as we were going about our business for several months, which, honestly was really fun because there's nobody around. It was great. We had good science to do. I was really enjoying what we were doing. We had funding coming in, and no one else around to bother me. It was, otherwise, if I didn't have to be on Zoom for most of my life, it would have been amazing. But it's been a really interesting change in the lab. I don't know if it's better or worse, but I'm still here, and we have funding for a while, and that's a good thing for coronavirus field in general, is that there's obviously more expertise and more understanding about these viruses being important to study, which I think is good in the long run.
Are you doing lab work still, Matt?
I am. Come on, you're not in the lab?
I just wander in and I'm told to leave.
No, absolutely. So I help out with mouse experiments and plaque assays, and we have a good structure in the lab now where people are working together and making plates and doing assays all together for everything we're doing. But I certainly step in and help out where I need to be for BL3 work. So I've always stayed in the lab. That's always been kind of what I did.
So you do real live, honest to God coronavirus plaque assays, right?
Yes, we do.
Isn't that wonderful?
Yeah. You don't use pseudotypes at all, right? You use authentic.
We don't. We don't actually have any pseudotypes in the lab.
Cool.
It's all live virus work.
That's good.
The new pseudotypes are incredibly useful. I don't negate any of the pseudotype work. I think again, there's no way to respond to this outbreak this year without all of these pieces together. And so there's been an amazing utility and really collaboration across all kinds of spectra. A lot of people came into the field and they'll leave right away when their grant doesn't get funded, and that's okay. That's just, it's the inherent part of an outbreak. It's the way it works. And there's good data out there and there's not good data, and that's okay. We'll figure that out as we go. But you can't, there's no way we could have responded to the pandemic with, 10 coronavirus labs in the United States working on it. It's just not possible. So everybody needed to come in, and it's good. It's been a great experience to kind of watch all of this happen and to be able to play our part in the role that we could play this year.
So have you tried to think about where you see yourself or your lab or even the pandemic a year from now, 2 years from now? What's your crystal ball say? About what we're going to be working on? Or I think I'm interested in both. What's your vision of the state of the pandemic on the one hand? And I'm also wondering, once we get out of this, where does that leave you? Our coronavirus is going to stay on people's radar, or everybody's going to say, okay, we can do that now. Let's go back to normal. No, I mean, on that sense, I mean, we're a coronavirus lab, so we're not changing what we do. Right. There's certainly a lot to learn about this virus and this family of viruses. We have many more tools now than we did a year ago, many more fun little gadgets in the lab to play with than we did a year ago, and we're not changing what we do. I really think for at least science-wise, one of the big things we're interested in is really looking at broad spectrum antivirals. And there's a big pan-coronavirus vaccine initiative now. And I think that is really important to drive for the next one. And so after SARS-1, everyone, worked on SARS-1. They left the field and there was a handful of us left. I was a postdoc then. When MERS emerged, there's another influx of people. We learned a little bit more about coronaviruses then because we knew everything we did for SARS-1, we used for MERS. People left the field again. Now with 3 highly pathogenic coronaviruses emerging in 18 years, I'm pretty sure people realize this is something we need to do. But I think the really unfortunate thing is after MERS, we all said we really need to develop therapeutics for these family of viruses. And we tried for, grant application after grant application, trying to drive this home. We had several projects in the lab looking at broad spectrum antivirals, and so that's still ongoing. It's been a really great project, now funded by a company called Keto Pharma, who is helping us develop this family of drugs that we found. We published this paper last year, targeting the ski complex as a broad spectrum antiviral, and we're still really interested in that as well. And all of the other drug work we're doing outside the vaccine work, we have a large drug project through a DARPA funded project with Don Ingber and Ben Tenuver, Mount Sinai, and at the Wyss Institute. For Don, we're working with Gates Foundation and BARDA on really identifying these antivirals and anti-inflammatories that we then are trying to synergize together to make really things that can inhibit SARS-2, but also I think they're going to have broader aspects and really targeting the host response to the virus. And so I think all of these things together will help us in the future, and we're going to certainly stay in this field and push this along. So I certainly think that people realize now, especially funding mechanisms, that this is not a flash in the pan. And we certainly have to be prepared for the next one and the next one and the next one. We don't often learn our lessons very well. So I'm not super confident we're going to learn everything and remember everything from this outbreak. But I think people's memories are pretty good when they have to sit home for 6 months. And so I think it's going to be, in everyone's mind, at least for a while. So I feel pretty confident in kind of the direction that we're going.
So you're somewhat confident that we can have broad spectrum antivirals and maybe even vaccines to be ready for the next one? Because there will be a next one, right?
I think there certainly will be a next one. Whether it's another 8 years or so on this kind of cycle we've seen so far, or shorter or longer, I don't know. I certainly don't have any prediction there, but I think that we certainly have to be ready for it. And I think it's certainly possible to do this, whether it's looking at repurposed drugs and combining them together and finding ones that work against this virus and this family of viruses. So we can test all of them now, SARS-1 and MERS and SARS-2 in animals, and we can do these kind of combination studies between drugs and different viruses. That's really one of the driving forces of the lab now. But I think the more we learn about these viruses too, it gives us more ability to target new aspects of the cell that we can now look at the host side rather than just the virus side for targeting. And combining things that target the protease or target the polymerase of the virus, as well as things that either alter interferon response or alter pathways in the cell that are important for replication. All of these things are going to be important, and I think going to be the ability to target these for multiple viruses is clearly there. We just have to find them.
So can you tell us a little about your FDA-approved drug screen, and that's what's led into some of these future hopes and projects?
Yeah, sure. So again, this was a project that we published a paper early on repurposed drugs that, there was a lot of repurposed drug work at the beginning of this. The funny story for the repurposed drug side is that we did this for MERS and SARS-1 after MERS emerged. We had really nice collaboration with a group at the IRF at NIH and USAMRIID, where we did a big screen of drugs, and we identified about 24 drugs that work against SARS-1 and MERS. We then worked for about 3 or 4 years trying to get funded to study repurposed drugs in animals and develop them further against MERS and SARS and other coronaviruses, continually being told, no one cares about repurposed drugs. You really need to find your own drugs that are new, and this isn't the way to go. And then at the beginning of this pandemic, all of a sudden everyone said, we should screen these large libraries of repurposed drugs. Why aren't we doing that? And we weren't a high throughput setup at that time. We are now, but we weren't doing 15,000 drugs at one time as part of a screen. Our initial story, we went back to those same drugs for SARS-1 and MERS and looked to see if they worked against SARS-2, and the majority of them do. So they were all really targeting the host, and that then led into these other drug studies with Donenberg and Bentenuver, and then with other studies now with Gates Foundation, where we're looking at things that come out of multiple screens through different cell types, doing work with AOI cultures, doing transport systems, and now we're growing other lung organoid-like systems like this emulate lung on a chip system to really understand a bit more of the biology of these viruses, of these drugs, and how they can affect the virus and inhibit in a more in vivo-ish system, but still in vitro. And then that translates into the animal work, and then hopefully clinical trials as we get there.
I wonder, Matt, if you have some insight as to why. So, we did really well with vaccines, but the antivirals kind of never got anywhere, right? Remdesivir was a disappointment, and not much. I mean, there's one other, I think, molnupiravir. Is that it? That looks promising, but nothing has helped patients. Right? Do you understand why that might be?
Because they're hard. They're hard to find. I mean, they're difficult. They're not in the time frame that we've been doing this. Most of them are not drugs that are targeted, that have been directed to the virus. Yeah. We're doing screens on these all the time. I think translating in vitro screens into animal work has been difficult. We didn't have a drug previously that worked against SARS-1 or MERS either. So I think that they're just viruses and making antivirals are a hard thing to do, where you can balance toxicity to a human and an animal, as well as targeting the virus. The other thing is you have to target the virus early on. And so once the virus isn't in you and you're causing and replicating at high levels, you have to catch it early. And I think that's what's shown for remdesivir and EIDD 2801, this small whatever the Ridgeback Merck drug, right? I won't pronounce the name. Is you still have to catch them early on. And we knew this from SARS-1 and MERS. Yeah. Even if you do monoclonal antibodies, if you give it 2 days after infection in a mouse, the antibodies don't work. So you have to really catch it early. So I think that looking at both therapeutic treatment of people with these drugs is important, and running these trials as fast as we can in randomized control trials is good. But also doing prophylactic dosing. So once we have the number of cases down to a low enough level and someone in your family becomes positive, can you then start taking doses of whatever medicine we find for 2 weeks to eliminate spread, right? That's the other aspect of where we need to go. And so it's all layers, right? It's layering the testing to vaccination to all of these drug studies that are coming on. All of this is just the way we have to go through this. And I think, it's certainly the way that viruses work. We know that viruses are kind of amazing little machines, and they outsmart us at every step, and we have to figure out their ways around them.
You mentioned, go ahead.
I was just going to say, I just think overall about how many vaccines we have for viruses in general and what success there's been, and how many really good antivirals we have in comparison. It's, to me, it's not a comparison. There's just way more vaccines. So it kind of makes sense that antivirals are going to be hard all the time.
We have a lot against the viruses that infect you for a long time, like HIV and herpes viruses, right? But the acute ones like this are tough, as Matt said, because you got to get it. I mean, if you have 10 years to treat an infection, that's great. But if you have 7 days, that's tough. It's really
even
hard.
For HIV, right? A single one of those drugs isn't that effective. So you have to layer these on over time, right? And that's why it's cocktails.
Of course.
This is what you do. And that wasn't figured out in 6 months.
No.
That was years. I think that's just the way we are. That's just the way this works. And it's okay. This is what we're doing.
And that pandemic is still ongoing, by the way. HIV/AIDS.
It is.
Hasn't gone anywhere.
Hey, there's another issue that you can compare vaccine development with drug development, and that is drugs are used to treat diseases. Vaccines prevent diseases. So you can use a vaccine for everybody, basically. Using a drug is more targeted, and therefore you get less money back for your return, I think. And that maybe that's why there are fewer drugs than there are vaccines for viruses.
Yeah, I think that they're really hard to target, right? And I think the drugs are easier to evade as well. So whether you're targeting a protein, I mean, it's not like there's been no drug development for coronaviruses over the years. There's been a lot. Once you translate them to animals, not many work that well. Yeah. Remdesivir is one of the shining examples of that. And there's a lot of protease inhibitor work that's being done previously, and they can evade them pretty quickly unless you have a really, really potent inhibitor. So there's we're working with Pfizer on their protease inhibitor that works great and works in animals, and that's ongoing as part of this development process. So there will be things coming, and I think layering these drugs together is the next phase of this.
So let me ask you one more before we let you go. Tell us your view of the end of this pandemic. Is it imminent, or do we have a long way to go, you think?
I think both. I know that's not your answer that you want. I think that we're not answering anybody wants. I think so. Just yesterday, today's March 8th, so March 9th. So March 8th, CDC came out with guidelines saying that vaccinated people can hang out with vaccinated people without masks on and be inside. I certainly think that is the beginning of the next phase of this outbreak is with the number of cases going down, with more vaccines in arms around the world, we certainly will be able to get back to being around people again that are outside of our immediate family. And that's certainly good on social aspects as well as just humanity of this in general. I think that the big worry is about the variants that are out there that are kind of lurking in the background and or the forefront, depending on what country you're in. The ability for the vaccines to work against the UK variant, the B117, the original one, is really strong, which is good. But now there's a B117 with this 484 mutation, which is evading some of the vaccines. That's not great. And that's shown up a couple places in the United States, as well as the South Africa variant. So I think that we certainly, all of those are still protected at some level by the vaccines. So that's why the big push is to get, 3 million plus people a day vaccinated so that we can really get through, the next 100 days and get another 300 million people dosed. So that we can get as many people protected as possible. And so I think on the short term, the country will be opening up again, whether because governors just say we're open, or because of real science. But I think that we're getting to the point where it's safer to be around our friends and family again. And I think the other part of this is, will this become a seasonal virus that circulates and becomes like one of the other 4 seasonal coronaviruses? I think there's evidence that that's probably going to be some level of what happens. And there's certainly models that show that. I don't really, we don't really understand much about the seasonal coronaviruses themselves, about why we still continually get infected by them, every 2 or 3 years, even though we're all seroconverted. And there isn't really a lot of data on that. And that's certainly another aspect of this outbreak, which has been really enlightening, is the lack of information we have on those viruses. And so there's a lot to do, and it's okay. I got you. I have a pipetter and a lab and a BL3.
So the comparison, the thinking about whether this is going to become more like a seasonal coronavirus, and thinking about the comparison between SARS-2 and those viruses, it seems to me that a fundamental question is whether the virulence or pathogenicity of SARS-2 is actually any different than the seasonal coronaviruses, or if we're just in a situation where we happen to be have an older, naive population. Okay, so 2 ways of looking at that. If the existing coronaviruses infect an older person, do you have lousy outcomes? Or, the flip side is, will this become more like a coronavirus? Are there studies that compare the pathogenicity or virulence of the common cold coronaviruses with SARS-2 in a meaningful fashion?
Not per se, no. There really hasn't been. The part of it was because we didn't really have a lot of the cold coronaviruses around, at least this year, when people are really looking. But we certainly know from the immune response data that there may be some cross-reactive T cells between these, between people who are seasonal coronaviruses as well as against SARS-2 proteins. But I think the really big difference in this outbreak was that we all are seroconverted at some level against the seasonal coronaviruses. They don't have to be 100% protective, but we have it. Yeah. We're coming in where, the large majority of the world has never seen a SARS-like coronavirus before. And so you have a naive population. I mean, it's exactly what everyone teaches in virology classes, and we all give like pandemic virus talks all the time. It's the worst thing to happen is this virus to come in that is going through a naive population, right? It's every movie about a virus that you see, in the theater, or used to see in the theater, where this happens. And I think that's it's a textbook response. The interesting thing about this virus, though, as you've talked about before, is that for SARS-1, everybody who was infected within 24 hours or so, you had symptoms. And then during the 2003 outbreak, you got quarantined or you stayed home, you went to the hospital. And once they figured out that it was airborne or that was aerosol transmitted, then they used aerosol protection in the hospitals, and everybody was somewhat more protected. With this virus, there's transmission before you show symptoms, right? And so that is the big driver of all of this outbreak, and why we're walking around with masks, because we need to block those aerosol generation, of humans. Yep. And so it's a weird thing to be working on these viruses, talking about them for years, and then being in the middle of a pandemic with the virus you've studied for a while. It has really been a strange year. I keep saying that to all my friends and family, and no one gets it, but it's really weird. It really is so weird. But I at the end of this, I'm happy for the role that we're playing in this. I think there's good science coming out of the lab now. And I really love the collaborations we've had with all these companies and other academic collaborators. And, every bit of these stories, we learn more and more about these viruses. And so we're going to get through it. And we're almost there.
Matt Frieman, University of Maryland. Thank you not only for coming today, but also for your work. We appreciate it, Matt.
No problem. Thank you very much. And good luck, Matt. And stay healthy.
Thanks.
Yep. Bye-bye.
Thanks. This was great.
See you number 11.
Yeah.
All right. Bye-bye.
Bye-bye.
See ya.
Let's do some email here. I will take the first two short ones. Lisa writes, just watch Mavis, the structural maven, TWiV 448. My goodness, what a force. Thank you for remembering her and introducing her on your last podcast. What a smart, fun, resilient, and inspirational person. Wow. Thank you for all you do, Lisa. And Christiane. Hi, Kat. This was to Kathy. This is from Kathy's colleague over at University of Michigan, Christiane Wolbes. Just saw this email and thought it might be of interest to the TWiV crowd that has been lamenting the lack of T-cell analysis in COVID. There's now a commercial test to measure SARS-CoV-2 cells from a company called ImmunoSeq. It's mainly meant for research, I think, but it's good that there's a kit to do that. That's a good start.
Yeah, I sent it to all of you wondering what Brianne would think about it.
I was excited to hear about it.
You're going to buy a couple, Brianne?
I think that would be fun.
Sure, go for it.
Sure.
And check your T cells since you've been vaccinated. See if you have some virus-specific T cells.
Exactly.
Speaking of that, Alex Setti, I like to call him Alessandro, but everyone calls him Alex. It's unfortunate because it's Alessandro is a nice name. And Shankrati have published an interesting preprint on how the variants don't really impact T cell epitopes.
I saw that. That was also a fascinating preprint.
He's coming on in a couple of weeks. I asked him to come on and discuss that, and we can have a nice chat. And, you know, the funny thing, so I'm on this call every other Sunday, and he was on it, and he was asked to explain it in 5 minutes, and he did. And he said at the end, you know, unfortunately, it's a boring paper because we found nothing. And I'm like, no. It's important.
Yeah.
And so anyway,
I do not find it boring.
It's not boring at all. I don't know why he would say that, but it's really great. And so we will have him explain the potential implications of that. All right, Kathy, can you take the next one, please?
All right. Dear TWiV hosts, thank you for the interesting, illuminating, and useful information you provide. As a layperson with no background in virology or any of the other sciences most pertinent to the SARS-CoV-2 pandemic, back in March 2020, I learned a new word, virion. Since discovering TWiV a month or so later, I've listened to every episode and watched a few from the emergence of the new coronavirus to the present. Several weeks after becoming a listener to TWiV, I learned of Vincent's blog and read some of those posts, at least a couple of which, July 28, 2004 and July 22nd, 2010, explained the term virion. With that in mind, I've been surprised that the word virion is not used more frequently by hosts and guests on the podcasts. I have heard the word used several times on the podcasts, but not as regularly as I expected. Apart from the word virus itself, the other word that is used most often seems to me to be particle. Sometimes I think particle is used to mean virion, but other times it is used to mean a fragment of viral RNA or DNA, or other times to mean viral antigens. So I ask, why not use the word virion when that's what you mean, since it seems to me to be so clear and explicit? I also thank you for taking the time to define or explain the many words and concepts that probably seem self-evident to all the hosts, but which may be new or murky to us lay listeners. This is particularly useful to me when you delve into biochemistry. Several of the hosts provide this service, and I think that Rich is the leader. I do acknowledge that your audience has grown continuously, and probably some listeners miss some episodes, and it would likely be tedious, even to listeners, and impractical to define or explain every word or concept as it comes up. But every time it comes up. But I do very much appreciate the clarity you bring to the virology, immunology, epidemiology, and with Dr. Daniel Griffin, medicine around the pandemic. Sincerely, Earl. So now, Vincent, you're going to explain what is virion versus virus and how they're used.
So I have to say
Correct.
that, so when I joined the textbook Principles of Virology in the 90s, I learned a lot of things that I didn't know because, you know, there's no book of definitions in virology. If you know, even things like variant and strain and isolate, there's no place where you could go look it up. And virion, I learned, and this was Lynn Enquist and Jane Flint, they said that's the word we use to mean the infectious particle. because not all particles that are produced from a cell are infectious. And so you use virion to describe the ones that are. And I think we don't use it all that much because we never know what we're talking about. And so whether we're talking about infectious or non-infectious particles. So, for example, an inactivated virus vaccine contains virus particles, not virions, because their infectivity is gone. However, I must say, I think most virologists don't actually use it in that manner. And so few people use the word at all. And those that do just use it for everything. But yes, I think from my view, that's why we don't use it all that much. But yes, it should mean the infectious particle. I don't actually know if that is how the term was coined originally, right? But the thing is that when you say virus, that's an organism, right? With a virus particle and an infected cell phase. And most people, when they say virus, they're thinking of the particle, right? So I try and distinguish it in that way. Anyway, that's my 2 cents on that. I'd be interested to hear what others think about that.
A couple of things. First of all, if I look up virion, the first thing that pops up in Google is the complete infective form of a virus outside of a host cell with a core of RNA or DNA and a capsid.
Good.
Now, there are other definitions that I can run across that don't immediately make the distinction of infective.
Yeah, nice. Very good.
Personally, I did not have infectious or infective as a qualification for the use of the word virion. That's, I mean, nobody ever told me some precise definition of virion.
That's right.
And I think it's an interesting question, how one uses the word virion versus particle. Me personally, I don't use it very often, and usually I use it in the context of it's when I have a picture of a detailed structure in my head. Okay, when I'm detailing structural elements of this thing, I'm likely to call it a virion. And beyond that, it's all pretty vague. Obviously, I haven't had a rigorous definition, and certainly infectivity wasn't part of my definition.
No, as I said, it wasn't part of mine until I wrote this book, because Peter Palese never said, Vincent, virion is infectious part. He never told me that. And that's how we learn stuff from our mentors, right? And colleagues.
Yeah. Go ahead, Brianne.
Oh, I was just going to say, I think that for me, I use particle as a more accessible term. I feel like I use virion if I'm talking to people who have more virology background, and I use particle if I'm talking to students or people who have less background for clarity's sake. And I think that's the only distinction I've generally used. So I am happy to learn more about what I should be saying.
So I think I typically don't use virion very much because I've learned of this narrow thing that it has to be infective. And it's just safer and more general if you're going to talk about the virion protein versus the virus protein. Virus is going to work, and particle also works.
So you had the infective thing as part of your definition, Kathy?
Only since I've started using the Flint textbook. So that goes,
that's good.
Less than 10 years, probably about 10 years.
I mean,
3 editions.
I must say that Earl did his homework, right? He looked it up, and so it could be that some more people would be interested. But I think, as Kathy said, it's hard to know when to use. So let's say, there's droplets, respiratory droplets that go in and they deliver virus particles, because you don't know if some of them are going to be infectious and some of them are not. You can't say virions only, or I guess you could. Some of them would be virions, but yeah. Here in the lab, we use virion all the time, okay? And in my course, I use it a lot. But so that's the story anyway. But I think this whole idea that you learn from your mentors and they're not always right, it's interesting. The reason I mention it is because I recently wrote a blog post about strains, isolates, variants, and strains. And I had originally asked Jens Kuhn, who I think of as the go-to person for nomenclature of viruses, because he's on the ICTV, the International Committee for the Taxonomy of Viruses. So, in fact, he published a paper a number of years ago where he defines isolate, strain, and variant. And I agree with his definitions. But before he does, he says, you have to recognize that there's no one place you can go to find out the definitions of these terms. People learn from their mentors, and it could be right or wrong what they learn. And I think that resonated with me, because that's true. A lot of stuff I learned from my mentors was right, and a lot of stuff wasn't correct, right? So let me think of something. I can't think of it right now, but a number of things have turned out not to be correct. But I must admit that writing the textbook, we have 5 virologists sitting around a table, and you come to a consensus for how you should define things and represent things. And I didn't agree. I mean, we still disagree on how to pronounce the word that means programmed cell death. Right? Some people say apoptosis, and some say apoptosis. And we had long meetings where they called me unsophisticated for my pronunciation of the word. Okay, fine. Helicopter, but then I was told that doesn't matter. Anyway.
Or saponin versus saponin. I've always heard saponin, and saponin I think is the name of the tree, so maybe that's the way to pronounce it. Who knows? Vincent, do you think there should be a committee that's formed that rules over the nomenclature, so everybody uses the same words for the same things?
I don't think we need more committees. The funny thing is, in Jens's article, he said the designation of a strain can only be done by an expert international committee. And I thought, God, right. We'll never get there. It'll take forever.
No, look at how much trouble they had classifying Pluto.
So
that's a totally different field, but
And I will tell you, Dixon, if you asked my students about that and whether we should have a committee, they would be all for it.
Yeah.
I think that there are many times throughout the semester of immunology where they sort of throw up their hands and look at me and say, immunologists are no longer allowed to name anything.
This is true.
We're just
That's right. Exactly.
So, in the back of the textbook, Principles of Virology, there's actually a glossary where we define things, but they're not all there, of course. So some people do make an effort to define terms, and we work pretty hard on that glossary. But I think that it's kind of like the English language, right? It's a living thing, and it grows and adapts. And to some extent, I'm okay with that. But I do think in science, you have to be precise and acknowledge what you're talking about. And so in particular in a textbook, it's particularly important to have the same definition in chapter 1 as in chapter 25, right? You can't use different. And even when you have different authors writing them, they have to agree on the definition. And so that's why we would have these discussions and come to some kind of consensus. So,
right.
Fortunately, there's no pronunciation of apoptosis implied, so that doesn't have to go. All right, Rich, can you take the next one?
Suzanne writes, this is about Texas masking. One problem with dropping the mandates will be for businesses who want to follow the guidelines, having to deal with customers angry that we're not letting them fall back on their, quote, personal responsibility like the governor said, end quote. It's easy for Abbott to say businesses can do what they want, but he's not going to be the 18-year-old behind the register trying to hold firm against big, loud, selfish fools. Suzanne, yeah, absolutely. Now, I must say that I tread pretty lightly on this last time when we discussed it. The governor did say that masking is recommended. Okay. And he made this statement that I just, it was a little vague, but basically saying that, Texans don't need to be told what to do. They know to do the right thing, and masking is recommended. The implication of that is that everybody would just, stay masked up. But is that going to happen? I doubt it. And what Suzanne says is exactly right. And I think the only way as a small business that you can deal with this, at least, and I think some businesses are, is the same as no shirt, no shoes, no service. It's no shirt, no shoes, no mask, no service. Okay. And if somebody wants to get mad, fine. They can get mad and then go somewhere else.
As long as they don't hurt you, that can
as
happen,
long as they don't hurt you. Okay. But I mean, it is a little crazy. I mean, wearing a mask, what is the problem? Really? Think of it like shirt and shoes. Somebody going to come into your place bare-chested, okay, and start screaming at you because you won't serve them an ice cream? Give me a break.
Well, unfortunately, Rich, last year, I think it was a bar in Buffalo, New York. Guy walked in without a mask, and a customer said, "Please wear a mask." The guy knocked him over, and he died. He hit his head on the ground. I mean, this is right. You can't have this stuff happening.
Oh,
but I agree.
Right.
Anyway, Suzanne is an old listener. I recognized her email. She's in Texas, and first time she's written in a long time.
Well, it'll be interesting to see what happens. Let me see. What's today? Today's the 9th.
Yep.
Those new rules go into effect tomorrow.
All right. Dixon, can you take the next one?
I can. Stephen, or Stefan, depending on how you want to pronounce that, writes, Dear TWiV team, on today's episode, you discussed the status of Novavax vaccine candidate, which is behind Johnson & Johnson, despite phase 3 data being in press released around the same time as J&J. The short version is that Novavax's data was from a non-US, as you noted, trial that doesn't match FDA specifications. I know that the number of subjects is low, 15,000 compared to the 30,000 in the Pfizer and Moderna's trials and 44 in J&J's, and I suspect there might be other variations. They are proceeding with a US-based phase 3 trial that finished enrollment in February. That said, they're apparently in talks with FDA to gain authorization based only on the UK trial, and Novavax hopes that if that is allowed, they could get authorization in early May. But if they have to wait until the US data is available, it may take a few more months, at which point, if President Biden is correct, we'll have enough doses of other vaccines to cover everyone anyway, so it might impact EUA. However you read that. Thanks, Stefan or Stephen.
So, as Matt said, the U.S. trial is in progress. So, I didn't realize this. We should know this, that you need to have U.S. participants, right, for the FDA to give you an EUA, I suppose. All right, Brianne, can you take the next one?
Sure. Raphael writes, hello, all of you from TWiV. I'm Raphael from Belo Horizonte, Brazil. I apologize for however poorly I pronounced that.
No, that's correct.
Oh, excellent. And I would like to talk a bit and hear from you guys about my opinion on the 76% number for Manaus in 2020. And so this was the 76% seropositivity data that we've talked about before. To me, it's impossible. If you had that, it would mean 1.6 million people infected with around 4,000 deaths at that moment. That would be the lowest infection fatality rate in the world by far. A lot of the deaths counted, as in Manaus, were from people that got infected and sick elsewhere and then went to Manaus, since it is pretty much the only city in the state of Amazonas with ICUs or even good hospitals to begin with. And there's a local law for the epidemic that means if you die in Manaus, you'll be buried there. You can't transfer the body. Add to that, the healthcare infrastructure in Manaus is already insufficient. You will get an even higher number of deaths. Manaus has 24 hospitals, but some of those are small facilities, not capable of treating severe cases of anything, really. Reinfections are still very rare. Only 2 are confirmed so far, and there are only 20 more under investigation. Virology professor Julio Croda, MD, PhD from Oswaldo Cruz Foundation, went to Manaus, and he believes that Manaus is yet to reach 50% prevalence. He also considers that the population affected by this second wave is socially different from the first. In 2020, public hospitals went full capacity first and then the private ones. This time it was the other way around, meaning that now the upper class that managed to quarantine during the first wave got infected. Some good old shoe sole epidemiology, I believe. Here is a link to some of this analysis. It's in Portuguese, but I believe that using Google Translate will be possible to follow it. And I have not looked at this yet. But to me, maybe the main flaw to consider is the blood donors. They are not representative at this moment. As a regular blood donor, I can attest that. Imagine you are in an epidemic. Who do you think are the people that are going out and about and end up donating blood? A person that never had COVID-19 and is locked inside the house, or a person who has already had COVID-19 and is less worried of getting it? So to me, a high level of seroprevalence among blood donors is just due to the fact that people exposed to COVID will be the ones that are walking around more freely, doing things, including donating blood. Almost all people I know, including me, that are donating are people that had COVID before and are more confident to go around places. Of course, we know that it's not 100% safe to walk around even if you already had it, but people still need blood donation, as I needed after an accident, and someone needs to do it. I don't know if I'm right or if it makes sense, but I believe this is one of the very few lines of interpretation of this study. Thank you all so much for your work and to share in public platforms. I've been following TWiV and related projects from you all since the middle of last year, and it has been a fantastic learning experience to me. As I said, I had COVID-19 in October, November of last year, and I'm glad to be safe, and I'm sure that all I learned from you guys helped me a lot to understand what is going on during these challenging days. Cheers, from Rafael.
What a nice name for a city, Belo Horizonte, huh? Nice.
I think it's Horizonte with a more of a t to it. Horizonte.
Okay.
Yeah.
Yes, that is correct.
Well, I've always been suspicious of that number,
Yep.
but not known how to think about it. And I appreciate this perspective. I think this is a very interesting perspective on that. And Raphael, good work.
Really
I
nice.
like it.
Mm-hmm. Yeah, I like the point about the blood donors in particular, that may be a biased sample.
Yeah.
Yep.
Yeah.
I like that a lot.
I think this interpretation of I think the reinfection story is good because we're told, oh, there are so many reinfections, but apparently not that many. But also the death, as he said, the death rate is not consistent with an infection fatality rate. So really good.
Very good.
Really good.
And also the people go to Manaus and then they get counted as, dying from in Manaus.
Yeah, right.
So that.
All right, Dixon, I'm going to take you out of turn so you can read this next one because I know
I sat here in fear that you would be doing that, but I'll try my best.
But you like to read the poetry, no?
Yeah, believe it or not, I actually rehearsed this morning to see if I could get the cadence correct with the genetic code, but I'm not sure I've succeeded. So I'll give it a try. John writes, Dear Twivers, a retired MD who has been listening since March, a sponsor since late 2020. It's great to hear your discussions of the current status of the virus and the literature. Loved Dagfinn from Oslo's version of Mr. Sandman and Dixon's mellifluous renderings so much that I added a couple of verses attached herewith. I'm not gonna sing this. I'm gonna
try and sing it. No.
Really go
just
for
the
it,
intro.
The chorus.
Okay. I don't know if I can do this. ATGC. No, I can't do it. I can hear it in my head.
ATGCUACGATGC.
Very good. Yes. Very good.
Mrs. Lab Woman, send a vaccine. Boom, 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 boom. Inject mRNA to start the machine. Make my cells will make the spike and make antibodies to knock the virus out while I drink hot toddies. Get the first jab, a piece of cake. Okay, the second one is harder to take, but that's a neutralizing scene. Let's knock out old COVID-19. Yes, more. Ms. Lab Woman, virus is clone. I'll take a shot without a complete genome. Just make an envelope to bring DNA in. With just enough bases to stop the invasion. Virologists Vinnie Brienne and Richard Dixon and Shirley Allen keep talking up what's on the scene. Let's knock out COVID-19.
Dixon, that was brilliant.
That was
Absolutely
excellent.
brilliant.
I told you I rehearsed.
That was brilliant.
They left out Kathy. I'm sorry they left you out.
That's all right. They left out Alan previously,
Yeah.
so this time he got included.
Right.
He's got a PS here that says a smile is contagious
Yes.
too, which he attributes to S. pastis. Do you guys know who that is?
I don't.
Pastis. No, pastis is a drink.
No.
Pastis.
Steve Pastis or Pastis is, if I'm not mistaken, I'll look it up, is the author of Pearls Before Swine.
Huh?
You could
Comic
be
strip.
right. You could be right. But I think I choose the pastis French drink instead.
Nice.
You'd like it.
All right. Mark writes, Dear Vinny and the TWiVs, here is a haiku inspired by listener letters to recent episodes, thanking TWiV for providing scientific and medical facts. Facts, facts, just the facts. Fight bad opinions and lies. TWiV spreads just the facts. As I write this, it looks like the New York metro area is getting slammed by another cold front. Not anymore. That was 2 weeks ago. Would it be cruel and unusual punishment to tell you that days are sunny and temps in the mid-60s on the Fahrenheit scale here on the California Central Coast? No, because Rich is always telling us it's in the 60s or 70s, so it's okay. It's not a
He had a few
problem.
bad days. He had a few bad days.
Kathy, can you take the next one?
Sure. Judith writes, Dear superheroes of TWiV, thank you for being such a steady source of information about the pandemic this past year. I've listened to TWiV off and on since my early postdoc days, 2009-ish, but your work on SARS-CoV-2 has me hooked, and I haven't missed an episode since January 2020. This is my first time writing in. I'm just a viral immunologist, like Brienne, turned stay-at-home mom. My PhD is in immunology and molecular pathogenesis from Emory University. I graduated in 2008, and I did my graduate work at the CDC on the role of measles virus accessory proteins P, V, and C. I now live in New York City and have most recently worked at Weill Cornell Medical College on influenza vaccine research using a lentiviral vector system. Before I had my son in 2016, I had migrated over to the writing side of science and was a medical writer in an agency for a couple years. When he was born, I took some time off from science to raise him, and now also my daughter. But I've recently gotten back into science through a freelance science writing gig with a great little website called studyfinds.org. I look forward to the day that my kids are old enough so I can get back to work for real. Anyway, down to my question. I'm curious what your thoughts are about the new CDC recommendations regarding what vaccinated people can and can't do. My internal red flags went off when I read that the CDC now recommends that vaccinated people can be with low-risk unvaccinated people inside without masks. My thought is mostly directed toward the kids. Isn't it true that even though children seem to be at low risk of severe disease, they are not necessarily much less likely to be infected and then spread that infection to others they are in contact with, for instance, at school? Furthermore, when they are infected, they can still experience negative outcomes such as multisystem inflammatory syndrome, MIS-C, and post-acute sequelae of SARS-CoV-2 infection, PASC. Additionally, there have been a number of reports now on the large percentage of people, I recently read 1 in 3, who experience PASC after recovering from a mild or moderate infection with SARS-CoV-2. So even low risk isn't no risk, and outcomes that aren't death can still be catastrophic for some. My feeling about this recommendation from the CDC is that it is an effort to not dissuade people from getting vaccinated, but I'm curious about your thoughts. Thanks again for your smart and entertaining podcast. Don't change a thing. Dr. Mom, aka Judy. And it's 4 degrees Fahrenheit, cloudless and sunny in New York City. 45 degrees Fahrenheit, cloudless and sunny in New York City. So I heard the CDC recommendation differently I didn't hear that CDC said vaccinated people can be with low risk unvaccinated people.
Yeah, it's true. It's on their website today. Yeah.
Okay.
Fully vaccinated people can visit with unvaccinated people from a single household who are at low risk for severe COVID-19 without wearing masks or physical distancing. Yeah, I don't think that was in the original, but maybe I'm wrong.
Yeah, is that both sides don't have to wear masks?
Yes, it is.
Yeah, yeah, it's weird. I mean, that's updated yesterday. Was that? I think that's when they first announced it, right?
I think so too. I saw it on the news last night.
Yeah, because what I heard was vaccinated people can meet indoors with other vaccinated people.
That would make sense right? That's perfectly good. Yeah, I don't get why the unvaccinated.
Yeah, I think the stories that I all heard were talking about grandparents getting together with grandkids
Who can't be vaccinated, right?
Who can't be vaccinated, right?
Yeah. So I think I agree with Judy that, you could still get infected and it could be a problem. I don't know why I would rather have them wearing masks if they haven't been vaccinated. But they didn't ask me. What do you guys think?
Rich is
Dead silence.
Yeah, Rich.
Rich talking.
We've already done that.
It's it's complicated. They're trying to balance a number of things here.
Yeah, they are.
Yeah, that's right.
The social elements, I wouldn't be at all surprised. Now, I trust the CDC not to do anything that really doesn't fit well with the science. Okay. So I have a hard time believing that there's a primary motivation of not discouraging from people to getting vaccinated. But I wouldn't be at all surprised if a bit of that creeps in here. I mean, I've seen a lot of stuff recently about how if you tell people that they're still going to have to wear masks indefinitely once they're vaccinated, people are going to go, well, why bother, or something like that, that's going to be discouraging. So I can imagine, I'll bet you that in formulating these, that came up in the discussion, but I doubt if it's a driving force. Okay. I'm glad I don't have to make these decisions. Put it that way. This is complicated.
So, Rich, you're a golfer. Did you watch the golf tournament this weekend?
No.
Too bad, because everybody at the golf tournament was told that they have to, in order to be an audience member, you have to wear your mask. So everybody showed up with their masks, but they were wearing them on their chins.
Yeah.
So, it's hard to breathe through your chin. I've tried it, and it just doesn't seem to work too well. And if I could exhale through my chin, it would still be okay, but I can't.
Yeah, well, there's a problem. A golf tournament has a lot of beverage consumption involved with it. And
it does. But it was windy, but still, just wait till you see the NCAA playoffs, and you'll see what happens.
So the other thing is that I think a lot of this has to do with the evolving perspective on whether or not vaccinated people transmit disease. Mm-hmm. And it seems to me that the evidence is increasing that transmission by vaccinated individuals is a very low probability event. Okay. And my guess is that factors into this as well.
Yeah, I mean, there's some data, but it's not extensive yet. So we need more. If it were me, I would have said vaccinated people can have small gatherings in their home without masks, but they have to be vaccinated. And I think that sends a message that you don't have to wear a mask forever, right? That would be enough.
Smart. That's smart. That's very smart.
So I wonder, it seems to me it's going to be a long time before kids are vaccinated.
True.
Okay. So when do we drop the restrictions? Okay. Are we going to have to wear masks until everybody, including the kids, is vaccinated? Somehow I doubt that, because I think that's going to be a while. So do you wear masks until all of the adults or a large fraction of the adults are vaccinated? So maybe you're anticipating conferring herd immunity on the kids. I don't know, because that's not going to be perfect. The kids are going to get sick. And this raises the whole bit about whether or not this is going to become another common cold coronavirus again. Maybe the kids are just going to get it.
So the population of the U.S. is how many million?
330.
330 million.
All right. And so there are 300 million adults. So that's 90% of the population. If you get all the adults, I think that's enough to protect the kids.
Gosh, yes. Gosh,
and
yes.
so if it's true that by the end of May, all the adults are going to be vaccinated, I think you can say, take your masks off at that point.
I think what Fauci said is by the end of May, there will be enough vaccines, and that by the end of June, everybody will be vaccinated.
Oh, that's right. You have to get them in arms. I forgot that part.
Detail.
And which actually takes me back to a previous letter about the Novavax vaccine and whether it's sort of lagging behind in the EUAs is gonna be too late. We're going to need a lot of vaccine for a long time. The more, the merrier.
No question.
Right. And if we're going to need boosters and all of that.
Yeah.
That's right.
Yeah.
So I think third and second grade teachers are going to be doing this experiment. Because if they show up without a mask because they've been vaccinated, will they become infected from kids who are not sick but still can transmit the virus? Yeah. That's a good test,
teachers will all be vaccinated, right?
I hope so.
They're over 18.
That's what I'm saying.
Yeah.
So if they can show up without a mask in their classroom without risk of catching the infection from their students, I think they'll all be in favor of having classes back in session.
Mm-hmm.
Right.
No matter what.
Right.
I think so.
While I was trying to look up the CDC details, I saw something else, Rich, that says that the Austin City Council announced, and it looks like maybe about 3 hours ago, that customers will still be required to wear face masks inside Travis County businesses, despite Abbott's lifting the statewide mandate.
Ooh, that's interesting.
Yeehaw.
Yeah.
Because there'll be news about that, I'm sure. Because I thought that there was part of the Abbott thing that said that local governments couldn't countermand that.
Yeah.
Not that Austin wouldn't go ahead and try and do it anyway.
Right.
Okay.
It's like they have.
That's interesting. All right.
Yeah.
Rich, can you take the next one?
By the way, before I start this, I confirmed that Stefan Pestis, is that how you would pronounce it, Vincent, is indeed the author of Pearls Before Swine. Mm-hmm. Has become one of my favorite comic strips. It's really quite well done. Jeanette writes, I've had my 2 doses of the Pfizer vaccine, and I'm so excited and grateful that I'm done. But for some reason, I started wondering what would happen if I kept going back for more. What would happen to your body if you had multiple shots? Like, if you managed to get 2 doses of Moderna and 2 doses of Pfizer and a dose of J&J, would you get really sick or really healthy or turn into a mutant superhero? Or is it like taking too much vitamin C and your body would flush it out? Thanks, Jeanette. Well, I can't really answer that. I'm going to have a stab at it and then throw it off to whoever else, probably Brianne. I think you just basically max out, right? And you it wouldn't do any harm. And after a certain point, it wouldn't necessarily do you much more good.
Yeah, I mean, in theory, you could make, a secondary response the second time and a tertiary and a quaternary and on and on. But at some point, the benefit of that boost would really be negligible and you'd kind of be maxed out, just like you said. Maybe you'd depending on what is going on with causing the side effects, you might be getting more and more side effects each time. I've been debating with some people about immune mechanisms of side effects lately. And you need an answer to that before we could totally say if your side effects would get worse and worse. But other than that, I think it wouldn't really make much of a difference at all.
Well,
I just want to know how you got all those appointments.
Well, she's speculating.
Yeah, I know.
This is hypothetical.
But think about when you're vaccinated and then you encounter virus. You don't really know it. You do get infected, probably at a low level, and then you clear the infection. You get a little boost, and you don't even know it. So I would now, of course, the vaccines probably have far more antigen than this little infection, but I think it's fine. But you don't need to do it. You're going to get boosted over the next few years because you're going to get infected over and over, and you're never going to know it.
So I keep thinking about the fact, Vincent, in the preparation for this show, up in front, the notes, you had in there a paper about the effect of a boost of the Novavax vaccine relative to one shot on the shape of the immune response. And you took that paper out, but I read it and I thought it was fascinating. I really like that paper.
So I'd like to see that then.
So I had sent the list of questions to Matt, and he said he didn't want to. That was someone else's study, and he didn't really know anything about it.
Okay, so fine.
That's why I took it out.
That had a lot to say about what the effect of a boost is, not just on levels of antibody, but types of antibody, where they are, T cell responses. It spoke to the whole shape of the immune response, which I think is really interesting. So maybe we could pick that up again another time.
Okay. Dixon, can you take the next one?
Yes, sir. As I said, I am muted. No, I'm not. Mona writes, you were discussing mathematical logs on TWiV 728 with an interjection by Dr. Dupamier that there were dead trees. So I was reminded of this joke told to me by my aunt, who did understand logs. As you know, in the Bible, Noah and his family built a large boat, the ark, to save the animals from an enormous impending flood. He took pairs of animals on board to safety so that they would multiply when the land reappeared. After the flood, he found a spot to tie up the ship and release the animals. But Noah kept track of the animals' reproductive progress because his sons went out to check. One time, as they were walking around, they encountered a pair of snakes and said to them, "Why haven't you multiplied?" The snakes replied, "We can't, we're adders." This was the first year. The next year, Noah's sons built some picnic tables using the dead trees lying around so the men could eat their snacks, and some lean-tos for shade from the hot sun so they could continue their inventory more comfortably. And then after a while, they came upon the adders again. But this time, there were baby adders as well. The sons said to the adders, "Why is it that you adders can multiply?" They answered, "Well, ever since you put in those log tables."
Lovely.
Yeah, I have a quick one. Two anteaters are getting onto the ark, and they look around, and one says to the other, "Two ants? Two ants?" Okay.
I get it.
Okay.
That would go quickly. All right, one more from Brianne, please.
It went very quickly.
Sure. Meredith writes, "Dear Twivers, longtime listener, first-time emailer. My name is Meredith, and I'm currently a medical writer, but not too long ago, I finished my PhD in immunology and microbial pathogenesis at Weill Cornell Medicine in New York City. My wonderful PhD advisor was Dr. Sabine Ehrt, in whose lab I studied 2 genes of unknown function in Mycobacterium tuberculosis. Right now, I'm halfway through episode 728, and the question of why are all the mice female has been raised. I had to pause and write to hazard my educated guess. It may have to do with housing the animals with their safety in mind. For tuberculosis mouse infections, we use all female mice so that we can group animals together during the aerosolization process and while being housed without having to fear that they would fight each other or eat each other. I'd bet the TWiV t-shirt that I caught during a podcast recording at ASM Microbe 2018 that this is the case here as well. Thanks for all your great science communication efforts. I actually volunteer with an organization called COVID Act Now, running the Research Rundown newsletter, in which I frequently link out to the Twix family resources. Test negative and stay positive, Meredith.
Nice. I like that sign off.
Yeah.
Very good.
Me too.
Oh, this is so cool that Meredith got a t-shirt. I love hearing from these individuals. And maybe someday we can throw out t-shirts again. Sorry.
Yeah. No, I was just going to say that interesting that, they did all their experiments with female mice. And when we started doing things with mouse adenovirus, we did things with all male mice because A, that was what was in the literature, and B, they were cheaper. So
Yeah. We do have another one coming up for next time about why all females, but we'll have to get to it next time. All right, time for some picks, and let's start with Dixon.
Well, then, I have the New York Times online, so every morning when I wake up, I start reading the depressing headlines. Mostly they're depressing, but sometimes they're not. But this one was really not depressing so much as conundrum-ish. I know we've discussed some of this on TWiV for the last year, but look at the arrangement of the number of deaths per million and the median income of the country, and they line up almost. There's some exceptions, but they line up. All of the poorest countries seem to have the fewest deaths per million, even though they've got similar rates of infection. And the article then goes into lots of discussion about the age pyramid of those countries, meaning that, in a country that has a lot of wealth and has a lot of medical care, you have an older population to deal with. And the older population is much more susceptible to this virus infection for lethality than younger people. Everybody admits that. So that might be one explanation, but I thought it was remarkable to see how much money doesn't matter when it comes to dying from something. And that's the essence of why I thought this was a good pick, to just say, even though everybody can afford medical care, basically, well, that's not quite true, but it's mostly true. It hasn't deflected this curve very much, and that's been the frustrating part of it, isn't it?
Yep. Over at World in Data, which I picked a couple of weeks ago, I have a great graph of age versus deaths, and it is just a mountain. It's amazing.
Yeah, exactly.
Brianne, what do you have for us?
So I have a site that I've actually used in the past for HIV-related details, but I've been really impressed with some of the things they're doing with COVID-19. And so I have a link to the COVID-19 vaccine monitor. That is basically a whole bunch of public opinion sort of polls about opinions on COVID-19. And so I think that sometimes people have been thinking about who believes what or who wants to get a vaccine or things like that. And these are sort of, this is sort of some data where people have been polled on this to see what they think about vaccines. And so there's data about, percentage of individuals who want to be vaccinated. And they have done that in December, January, February. So you can see it over time. And then that broken down into different groups, both in terms of things like racial and ethnic groups, but also things like political orientation, or whether someone lives in a rural or urban community, and just sort of groups in general. And when I was looking at this particular vaccine site, I also realized that they had another link on the top with a vaccine monitor with all sorts of questions that they've asked people about where they would prefer to get their vaccine, who do they think it gives them the best information about vaccines, and things like that. And I just thought this was really interesting data to take a look at. Although I will admit the first time I saw it, someone was sort of excited about the percentage of people who wanted to get a vaccine. And then when I looked at the data, I was a little saddened that the numbers were not nearly as high as I was expecting from the first thing I'd heard.
I don't understand. There's this one graph that shows it's politically driven. My goodness.
Yeah, that I just don't. I kind of get it, but I don't get it at all. It just, come on.
Yeah, that's cool. I was thinking we have so many cool resources that we share here on TWiV. I can never find them when I need them.
Yeah.
Brianne, do you know what KFF stands for?
It stands for the Kaiser Family Foundation.
Okay.
And I've tried to look into the background of them in the past when I've used their HIV data, and I haven't found as much as I perhaps would like.
Okay.
To tell you much about them, but they have been making public opinion data on HIV that I've been using for my students for a long time.
Excellent.
Rich, what do you have for us?
Well, it turns out to be, I think, at least modestly appropriate. I have an older article, I think it was from 2017, from the Smithsonian Magazine, titled "The Woman Who Stood Between America and a Generation of Thalidomide Babies." And this is about a topic that we've talked about on a couple of occasions in the past, about how thalidomide, which at least the older in the population will recall, was a drug that in the 60s was available by prescription for a variety of different problems, but it was prescribed for pregnant women to help them deal with morning sickness, which happens mostly in the first trimester of pregnancy. And it turned out ultimately that it was teratogenic. That is, it caused really horrific birth defects, typically very, very short arms and legs, and it caused a number of abortions and stillbirths as well. But it was approved in Australia and Europe, and it was not approved in the U.S. because of this woman whose name, let me get it, Frances Oldham Kelsey, who was working at the FDA at the time, and was responsible for reviewing the data. And she said, there aren't enough data here.
It's great.
To approve this. And in particular, I want to see studies in pregnant animals. And I want you to explain this neurologic side effect and et cetera. And so it wasn't approved in the U.S., and it probably saved us from thousands of these types of tragedies. So the article is quite good, and I also put in a wiki link about Frances Oldham Kelsey that gives a little more on her background. She's a fascinating woman.
It's great.
And I want to use it not only to celebrate her and that story, but to point out, I mean, we were just talking in this episode about how the U.S. has certain criteria for saying yes to a particular drug. And there are times when people say, well, it's being used elsewhere. Why can't we use it in the U.S.? Well, this is part of the reason. And Frances's efforts actually were at about the same time that the FDA was reviewing its policies, and a lot of her work, both indirectly and directly, ramped up the FDA's safety policies, and et cetera. So quite a remarkable woman and quite a remarkable story.
It's great. It's a great story. Great picture of her with President Kennedy honoring. It's just great. I love it. Wonderful. Kathy, what do you have?
I have just a pretty simple astronomy picture of the day from February 2nd. So I saved it for a while. And it's a picture of this quadranted meteor. And it's amazing. The meteor is just this streak across the field of view, but it's got all these different colors, and it explains where they come from. Blue-green from magnesium, calcium. There's some radiating violet, and nickel glows green, and there's red. It's just beautiful. And
it's a flame test.
Yeah, it's exactly what I thought of. Exactly. And I'd never heard of this, the meteors, the quadranted meteors, but they're evidently a January group of meteors that. So like Rich, I put in the wiki link to it so you can check more out about them.
It's interesting that along this line that you have segments of color. It implies to me that the elements that are giving rise to the different colors are layered in the meteor. Does that make sense? So you burn off one layer after another and get different colors?
You got it. That's true.
Is that at the burning of the meteor itself, or
yes?
Okay, not what it's passing through.
I mean, most of them are so small they disappear because they're the size of marbles or even less than that. Anything bigger, it would reach the surface of the Earth, and not all of it would blow up. So, what you're looking at are really absolutely small particles of stuff coming through.
Yeah, they call it radiant grit. So yeah,
radiant grit.
Nice. I love the name Quadranted. That's so cool.
Yeah.
Nice. All right, my pick is a book sent to us actually from a listener. I got it from Amazon. It comes with this little note, a gift from you, and then the who gave it to us wrote a little note. TWiVers, you have discussed attacks on science. You mentioned the dearth of expertise, but this book by scientists nails it. Please note, if nothing more, in the preface, page 10, the Sokol hoax from your avid fan, Vicky in Duluth. So here it is, Higher Superstition, the academic left and its quarrels with science. Really cool. So if you like the dearth of knowledge, it's all about attacks on science. So here in the intro, this is from 1998. As for anti-science, that's a very old story in our culture. In its accustomed forms, it would hardly have stirred us to take the trouble to study and write about it. The writing of Higher Superstition was undertaken only when it became clear to us from separate but remarkably similar experiences at our respective universities that something new and unwelcome had found its way into the academic bloodstream and thence into lecture rooms, journals, books, and faculty chit-chat, the systematic disparagement of modern science. A public response was clearly needed. So it's good. It's all about, this anti-science. It's the history of it and what's going on and so forth. So it includes gender-based stuff as well. So
excellent.
The Higher Superstition, if any of you would like to have it after I'm done, I'll be happy to ship it because it was given to all of us. Unless you wanted, we could rip it into 5 pieces. And each read a part of it. All right, that'll do it for TWiV 729. I'm saving that other pick for Friday, Rich. It's very cool, though. You will like it. Don't look at it right now.
Okay, I'll shut it down.
Yeah, you could look at it because you'll probably buy one of the things by Friday. That's so cool. TWiV729, microbe.tv/TWiV. Email goes to twiv@microbe.tv. And if you'd like to support us, we'd love to have your support. Microbe.tv/contribute. Dixon de Pommier is at trichinella.org, thelivingriver.com. Thank you, Dixon.
Thank you, Rich. Thanks, everybody. No, I wasn't going to say Rich. It was a rich experience. How's that?
That's good. You got out of that one. Very good. Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Which Austin may be seceding from Texas. Who knows, right? They like to stay weird. Someone from Austin recently, I don't know, talked to me and they said, Austin is weird. I said, yeah, I learned that. And they like to stay weird.
They vote Democratic.
Kathy Spindler's at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Brianne Barker's at Drew University. BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back in a couple of days. The TWiV is viral.